What up everyone, it's your boy Mr. Jennings here today with Big Orange Biology, and the main focus is on Mendel and his marvelous. So Mendel was a little Austrian monk who studied pea plants, and the big question usually deals with Mendel was the idea of why was he studying peas in the first? Well, first and foremost, sorry for the rough edit there, first and foremost he studied pea plants because, well, they were very easy to grow. The pea plants also were easy to mate. Not only were they easy to mate, you could mate them under very controlled conditions. They had lots of offspring. They had obvious traits. In addition to all that, they were also had very quick generation times. You can grow generations of peas in, well, a relatively short amount of time. Now, one thing you need to keep in mind when we talk about Mendel, we need to talk about why he was doing what he was doing. He was studying pea plants, studying reproduction. Time frame. In his time frame, the leading hypothesis was the blending hypothesis. We're going to make a note here. This is very flawed, but the idea was that traits blended from each parent. So each offspring had a blending of each trait. Now there's obviously some major problems doing that. Over time, eventually you'll just get a mix of everything, so there would be no pure traits. So what Mendel had to do was he designed an experiment to really test this and actually study how plants like the pea plant. So Mendel had a very simplistic experimental design. The very first thing Mendel did was he established true breeding plant lines. So you're going to see that word true breeding a few times. True breeding is another fancy way of saying, so if you got a trait that is completely pure, true breeding, that's all it's going to produce. That's the only gametes it can produce. It's going to be homozygous for whatever trait. Second thing he did was he started crossing the traits. So one thing he started doing was start crossing the purebred traits, or purebred lines, I should say. So he crossed those two different lines that were purebred, and he started seeing different results. And at the end there, what he ultimately did was he would then count the offspring that showed a particular trait. Various organisms had different little lines here, but his study was very simplistic. He looked at three basic things, and he got some very interesting data. So first off, he noticed ratios. These purebred lines, we're going to call them the F. So we're calling them the F1 generation. The purebred lines, when they actually did their thing, they only produced offspring. Showing only. And these offspring were called the F2. Then all he saw was the F2 generation produced 3-to-1 ratio with the hidden trait being the 1. So what he saw was out of 4 offspring, only one of those offspring would have the hidden trait from the previous generation. So he took 2 purebred lines, crossed them over, and he got an F2 generation. Once he crossed the F2 generation with itself, he found that 3 out of 4 had that same trait that the parents had, but 1/4 of them had a trait that was only seen in the true breeding, what we later known as recessive. So this led Mendel to come up to some major conclusions. First off, he discovered that there's 2 alleles. So there's always 2 versions of any one trait. And he called those versions alleles. And he determined that 1 allele came from each. So each parent gave 1 allele to their offspring. Also, he discovered that there's this thing called dominance. And that is, if there's 2 alleles, one will always be expressed. Now that be expressed here is what we would call dominant. And the other would be hidden. And we call that hidden trait the recessive. So he came up with the basic principles of dominance and recessive and alleles. And that's really the fundamental thing of Mendel. Now, it should also be noted, this is a modern adaptation and modern inference of his results. We know a lot more now than Mendel learned even then. Mendel's time, they had no concept of the idea of DNA, really, and the idea of chromosomes were far few removed. It wasn't until Thomas Morgan Hunt came along that we actually started learning more about it. All right, so we go on down here. Okay, now that comes up, Mendel came with two major laws. Laws of segregation and laws of independent assortment. So these 2 laws are what Mendel is really known for, and we talk about them a lot. And these 2 laws help exemplify the concept, are exemplified in the concept of. So law of segregation. Law of segregation here is basically that only 1 allele for a trait goes into the gamete. So when a cell undergoes meiosis and produces gametes, only one allele is going to go into that gamete. And this is a random process. Now, the randomness comes from meiosis. See, in meiosis, specifically metaphase, anaphase of both meiosis 1 and 2, specifically anaphase 1 and metaphase. Specifically in these events, the lineup is random, and the actual separation that happens in anaphase is the segregation. So the randomness comes from meiosis. So in specifically metaphase 1, when metaphase 1 happens, the homologous chromosomes are going to line up down in a homologous pair, and when they do so, it's randomly. They're going to go in different sides, they're going to flip-flop around, it's completely random. Then you have the homologous chromosomes separating in anaphase 1, and that is the actual segregation. They're going to opposite sides, they've segregated from each other. Now, conversely, we get the law of independent assortment. Now, the law of independent assortment basically is separate alleles, separate alleles for separate traits, spelled that, let's roll with that, are passed down independently of each other. So basically, the idea is one trait doesn't, specifically it's inherited. Now, as we're going to discuss, this whole concept has a little asterisk, and that asterisk is only. So we're going to talk more about linked here in the unit. We're going to talk about non-Mendelian genetics later this week. But we have to talk about Mendelian genetics, and that's linked genes. That's an example of one of those. And we're going to talk about more of that. So it's basically genes that are connected to one another, and they're inherited together, so they don't have the independent assortment. But more often than not, most genes are inherited independent of one another. The one gene does not influence the inheritance of another gene. That's the main overall. So at the end of the day, when we look at Mendel, the big thing is that Mendel's studies are really simplistic. He studied a pea plant. He had some very good luck in what he picked. It turned out he picked a really good model organism to help him develop these theories. And what we found is, while not everything's Mendelian, as we'll learn, it still gives a good foundational piece to understanding all the other variations of inheritance and how overall complex it is. So if you were to take away something here for the College Board, is one, the experimental design, identify the aspects of experimental design, but also the laws. So you need to understand the law of segregation and the law of independent assortment. Understanding those two laws and how those two laws can be broken, or how you can go against those laws, is really a good fundamental understanding of how biology really has exceptions to some rules. And we're going to find these throughout time, and that's really where the interesting parts of genetics come out. Complexity is really nice. All right, that's it for today. Hope and